mechanism of evolution? What is the origin of this variation and how does speciation occur? Even though Mendel had talked of inheritable factors influencing phenotype, Darwin either ignored these observations or kept silence. In the first decade of 20th century, Hugo de Vries, based on his work on evening primrose brought forth the ideas of mutations, large difference arising suddenly in a population. He believed that it is mutation which causes evolution and not the minor variations heritable that Darwin talked about. So, mutations are random and directionless, while Darwinian um, variations are small and directional. Evolution for Darwin was gradual, while De Vries believed mutation caused speciation and hence called it saltation, single step large mutation. Studies in population genetics later brought out some clarity. The Hardy-Weinberg principle. In a, in a given population, one can find out the frequency of occurrence of alleles of a gene or a locus. So, this frequency is supposed to remain fixed and even remain the same throughout the generations. So, Hardy-Weinberg principle stated it using algebraic equations. So, this principle says that allele frequencies in a population are stable and is constant from generation to generation. So, the gene pool, total genes and their alleles in a population, remains a constant. This is called a genetic equilibrium. So, sum total of all the allelic frequencies is 1. So, individual frequencies, for example, can be named P, Q, etc. In a diploid, P and Q represent the frequency of allele A and allele A. The frequency of AA individuals in a population is simply P square. This is simply stated in another ways, that is the probability that an allele capital A with the frequency of small p appear on both the chromosomes of a diploid individual is simply the product of the probabilities, that is p square. Similarly, of small a is q square of AA, pq 2pq. Hence, p square plus q square plus 2pq is equals to 1. So this is a binomial expansion of p plus q ka whole square. So when frequency measured differs from expected values, the difference means direction indicates the extent of evolutionary change. Disturbance in genetic equilibrium or Hardy-Weinberg equilibrium, that is change of frequency of alleles in a population, would then be interpreted as resulting in evolution. So 5 factors are known to affect Hardy-Weinberg equilibrium. These are gene migration or gene flow, genetic drift, gene migration or gene flow, genetic drift, mutation, genetic recombination, and natural selection. So when migration of a section of population to another place and population occurs, gene frequencies change in the original as well as in the new population. New genes or alleles are added to the new population and these are lost from the old population. There would be a gene flow if there is this gene migration happens multiple times. If the same change occurs by chance, it is called genetic drift. Sometimes the change in allele frequency is so different in the new sample of population that they become a different species. So the original drifted population becomes founders and the effect is called the founder effect. Now microbial experiments show that pre-existing advantageous mutations when selected will result in the observation of new phenotypes. Over few generations, this would result in speciation. Natural selection is a process in which heritable variations enabling better survival are enabled. to reproduce and leave greater number of progeny. A critical analysis makes us believe that variation due to mutation or variation due to recombination during gametogenesis or due to gene flow or genetic drift results in changed frequencies of genes and alleles in future generation. Coupled to enhance reproductive success, natural selection makes it look like different population. Natural selection can lead to stabilization in which more individuals acquire mean character value. Directional change, more individuals acquire value other than the mean character value. Or disruption, more individuals acquire peripheral character value at both the ends of the distribution curve. So this is the diagram basically of the three.